All right, so now we're on to the second part. We're going to today look at protein synthesis. So this is what kind of can also happen within the cell. You guys should know your functions of proteins. We said proteins have various functions within our body. They build our muscles, our antibodies. They help with your skin, hair, nails. They help transport your hemoglobin. So multiple uses throughout the body. The front page of your note sheet that we went through talked about the structure of proteins and how they're composed of amino acids. So amino acids, there are 20 different amino acids. They all have basically the same common structure, and we can combine them together. There's 20 different amino acids and long chains connected by peptide bonds to make unlimited number of proteins. So we want to look and see how that process is done, because we know that DNA codes for everything about you guys. However, DNA is located in your nucleus, and your proteins are composed out in the cytoplasm. So today we have to rely upon a little bit of our RNA molecules. You have messenger RNA, ribosomal RNA, and transfer RNA that you guys are going to be looking at. So I'm going to go back to the same area that we looked at yesterday with the nucleus, and I still have my strand of DNA. I'm only looking at a segment of this DNA. This is known as a gene. The code within this segment of DNA codes for a certain protein or trait. So when I go through this process of transcription that happens within the nucleus, what's happening is I'm going to take this DNA and now I'm going to be making it into messenger RNA. So while all of my RNAs are made with inside the nucleus, the nucleolus, and kind of travel out, messenger RNA is really doing its job inside the nucleus. The others we're going to see out in the cytoplasm. So we're taking DNA, making the messenger RNA. Remember our differences. DNA is double-stranded. That's why it can't get out of the nucleus. RNA is only a single strand, so it's a lot smaller, and it can fit out the nuclear pores to get out to the cytoplasm to take DNA's code in order to make proteins for us. Another difference that you guys are going to see is that we have thymine within DNA. However, in messenger RNA or transfer RNA, we're going to have uracil. Another one of our differences is our sugar. You have deoxyribose, and in RNA, you're just going to have ribose. So 3 differences that you guys should see along the way: single-stranded, double-stranded, your sugars, as well as your base pairs there. So if we look at this DNA molecule, we need to transcribe this into messenger RNA. And what that means is kind of put it into RNA's own language that it understands. It doesn't really understand this DNA code that we have here. So the same thing's going to happen that we looked at with replication, meaning helicase is going to come in, it's going to kind of unwind it, and it's going to open up that DNA molecule once again, just like it did for replication. It's going to get read and made the same way that we did for replication. So DNA is still going to get read 3' to 5'. So you'll notice my strands, they're anti-parallel. They're getting made in opposite directions. And my messenger RNA is also going to get made 5 to 3, just like it did before. So DNA is going to get read. And what's doing the reading here now, instead of DNA polymerase, since I'm making RNA polymerase is my new enzyme. So he's going to come on, he's going to attach, and he's going to start adding in my base pairs. And the first base pair he reads is adenine. Remember, I can't put a thymine here. There's no thymine in RNA. So uracil is going to replace it. And that strand is going to get made 5 to 3. So it's going to be made in this particular direction. On my other side, the same thing's happening. DNA is getting read by my RNA polymerase, and it's going to be made 5 to 3. Once it hits the end of that particular gene, the RNA polymerase kind of breaks off and my strand is complete. So now this single strand of messenger RNA kind of breaks off. At that point, my original DNA, it's done. I only needed that little piece copied. It's going to go right back together again. It's going to become that double helix, and it's just going to kind of hang out until it's called upon next time. At that point, this single-stranded messenger RNA leaves the nuclear pores, the opening within the membrane, and it's going to enter into the cytoplasm. When it enters into the cytoplasm, it's going to start the process of translation. So let's say the rest of this tabletop is translation, or is in the cytoplasm where translation takes place. Out in the cytoplasm, I have a ribosome. A ribosome is made up of 2 subunits, a large subunit and a small subunit. I also have let's go back to the ribosome. Ribosome is composed or connected together by what is known as rRNA, or your ribosomal RNA. So there's your second type. My third type is my transfer RNA, and that's these little blue structures out here. You're going to notice nitrogen bases on them as well. So in DNA, we said every 3 base pairs, TAC, AGC, whatever it may be, every 3 base pairs in DNA were triplets. Every 3 base pairs you're going to see here are known as codons on this messenger RNA strand. So I have AUG is a codon, CCG is a codon, and it keeps going. On my transfer RNA, every 3 base pairs is called an anticodon, so AUA. And what you're going to notice is that my anticodon is going to be complementary to my codon on messenger RNA, and that's what we're going to see. Also, my yellow structures are my amino acids. 20 different ones. You guys only have 4 that you see here. So we're going to kind of be transferring them to hook up to my messenger RNA strand to put together my protein. And that's going to be the job of my transfer RNAs. So as messenger RNA entered into the cytoplasm, it's going to activate or find an activated ribosome, and it's going to enter into the ribosome 5' end first. So my 5' end is first. I may have hundreds and hundreds of base pairs before that enters in, but until that ribosome, kind of like a little decoder ring, until he reads AUG, which is known as a start codon, nothing's going to happen. As soon as he reads AUG, it kind of pauses there at that first spot on the ribosome, which is known as an A site. We're going to start this process called initiation. So initiation is the first step of translation, and what that means is stop codon has been read. It's going to start calling out, the ribosome is going to start calling out, looking for the appropriate transfer RNA to match up with the codon. So AUG in messenger RNA is going to be complementary to UAC. So A goes with U goes with A, G goes with C. Here's my transfer RNA for UAC. Well, at that time, UAC is going to go find his appropriate amino acid. And they are going to be bonded together. They're going to be activated with a little bit of energy. And he's going to be kind of like a little taxi cab. He's going to transfer him around in the cytoplasm, and they will attach together. Codon, anticodon, with this little amino acid up at the top. At that point, everybody slides down from the A site, which is the activation site, to my P site, which is called my peptide site. So everybody slides down a little bit, and now my A site reads my next codon, CCG. Then he calls out for the appropriate anticodon. C goes with G, C goes with G, G goes with C is the anticodon I should be looking for. My transfer RNA, his little code tells him what amino acid to pick up. He can only pick up specific ones, and they're going to come in and they will attach. At this point, my 2 amino acids are now going to bond together. So if you remember the bond, dehydration synthesis, we're going to go through, remove some water, and we are going to create a peptide bond. And the peptide bond is going to join my 2 amino acids. These 2 amino acids together is known as a dipeptide. The ribosome can only hold 2 of these structures at a time. So everybody kind of shifts down. What was in the A site goes to the P site. What was in the P site now gets pushed off. But he leaves behind his amino acid because they're bonded together with the previous, or the amino acid after it. So this transfer RNA goes back out into the cytoplasm, and he's going to go try to find the same exact amino acid to attach to. So now my next one keeps going through. I have my next codon, which is UAU. So I have AUA. We have another one. It comes through. We have another peptide bond. Now we have this polypeptide. This step is my second step of translation. It is known as elongation. That's because I'm growing my polypeptide chain. I'm adding amino acids, and they're joining together with peptide bonds. This process is going to keep going, keep sliding down. He breaks off again. He leaves behind his amino acid. My next one comes in, ACC. I have UGG. We bring in my next particular amino acid, still growing that polypeptide chain. Eventually what's going to happen is that I'm going to hit a code known as a stop codon. That stop codon initiates my third step of translation, which is termination. So here I have a stop codon, UAA. It is one of 3 stop codons, UAA, UAG, UGA. As soon as the ribosome reads that, there is no complementary anticodon. There is no amino acid that will match with that. So it knows that, hey, that's the end of my protein. What I have, I'm good with, and everything kind of will go back out. My transfer RNAs will break off. They'll go back out into the cytoplasm. Messenger RNA can do one of 2 things. If he needs to make more of the same protein that we're going to see here, He's going to go find another ribosome and make more of it. If not, he'll kind of break down and get recycled. My ribosome, the two parts will break apart because he no longer has to be activated, and he's still within the cytoplasm. Now what's left is my protein. Now remember, amino acids, you need about 50 of them to make a protein. That's your smallest protein. They can be thousands and thousands of these amino acids put together. So the next video that you guys will see a little bit later on, we'll talk about now what happens to this particular protein within your body. Where does it go? How are we able to utilize it? So that will be the next step. Hope that helps.